Hi everyone, and welcome to our video on the cell cycle. This is part of our module 2 part 6 series looking at cell division, cell diversity and cell differentiation. And in this video we're going to be having a look at specification references 2.1.6a, the cell cycle, and 2.1.6b, how the cell cycle is regulated. First thing then is to make ourselves familiar with the little diagram of the cell cycle itself. So what I'm going to try and do through this video is show you a couple of different versions of this, just so that you are aware of the different ways it could be represented. It's not an exhaustive list clearly, but if we see it in more than one format, hopefully we'll be able to interpret any version you may see on an exam paper. So what we've actually got then is this little diagram at the bottom. It's generally a circle of some form, is showing us the cell cycle itself. And what we're going to see is we've got obviously interphase that makes up a large proportion of the whole cycle, and then mitosis, only a very small part of it. And if we have a look, we can see that interphase is actually divided up into these three other phases, G1, S and G2. So what we're able to do is in this video have a look at that whole process of the cell cycle in general. We can look at some of the checkpoints that are used in order to control this. And in our later video, we'll have a look at the process of mitosis in more detail itself. So first thing to remember is that when we're actually talking about mitosis, that is the division of the nucleus. So the nucleus is only going to be dividing during that M phase or the mitosis phase. For the vast majority of the time, our cell is actually in interphase. And if we look under a microscope at cells in interphase, it might appear to be quite uneventful in reality. But as we've now got a greater understanding of what's actually taking place throughout that process of interphase, then we do know that there's a lot of things happening in the background, and it's all to do with preparing that cell for that process of division that's going to occur in that M phase, the mitosis phase. So when we consider our interphase, there are those three parts, G1, S and G2 phases. We will sometimes see this phase referred to as G0 as well. Now, quite simply, this G0, G0, then that is going to be an occasional occurrence. Not all cells will have G0, but if they do, then this is where they're going to undergo differentiation or apoptosis or enter senescence. And what we mean by those three phrases are these. So differentiation, something we encountered back at GCSE, the process by which our stem cells become specialised into those specialised types of cells. Apoptosis is the death of cells, and we actually need cells to die. Sounds cutthroat, but we do, because when a cell reaches the end of its useful life, we need to get rid of it. So apoptosis is cell death. And then senescence is where our cells are going to stop dividing irreversibly. So basically they're going to enter this state of permanent arrest. So they're not going to die, but they're also not going to do anything. They just are. They exist. They're not growing. They are just there. So differentiation, apoptosis and senescence. If we think about that whole cycle, then we've got to have some method of basically checking it's doing what it should be and bringing about corrections if it's not. And so what we're going to use to control this interphase are checkpoints. And we've got two main checkpoints to be aware of. The G1S checkpoint, which is also known as a restriction point, and then we've got the G2M checkpoint. So they're the two main ones, but we do have other checkpoints along the way, like there's some halfway through mitosis, etc. And we'll look at a few of these examples as we go. But if you can only remember two checkpoints, the G1S and the G2M are the ones to remember. So what do these checkpoints actually do then? Well, first thing, quite important, they're going to prevent this uncontrolled cell division that would lead to tumours. So hopefully we're making that link there, something that's caused by tumours, cancer. That is where we've lost this control. So something has gone wrong in the control of cell division. Therefore, our cells are just happily dividing as many times as they fancy, and therefore we're forming this mass, this tumour. Second type of checkpoint that we will have is to detect and repair the damage to any DNA, because the replication process is not perfect. Third type is to prevent the reversal of the cell cycle. And the last one is to ensure that the DNA is only duplicated once per cell cycle. So it's kind of avoiding that build-up of excess DNA within a cell, which would then interfere with its functionality. What we're going to do then is we're going to go through and have a look at each of these phases and the events that take place, and then also some of their checkpoints. So the first phase we're going to have a little look at briefly is the M phase. So in M phase, this is where our cell growth has stopped and we have mitosis occurring. And remember, mitosis literally defined is the division of the nucleus. We're also going to see cytokinesis, which is the division of the cytoplasm. So in our M phase, no cell growth, nuclear division, and cytokinesis, cytoplasmic division. In terms of the checkpoints that we see in our M phase, we are going to have a checkpoint that triggers the condensation of chromatin. So remember, chromatin is the material that the chromosomes are made from. And halfway through our little mitosis phase, then we're going to have a metaphase checkpoint. And that's just doing a little check to make sure everything is there in place and as it should be, ready to complete mitosis. So we've got those two little checkpoints in our M phase, one at the beginning there, triggering that condensation of chromatin, and another one halfway through. Second phase we're going to look at is our G0 or the gap phase. And the events that happen here, as we've already mentioned, apoptosis, differentiation, or senescence. And do remember that this isn't all cells that will enter this, and some types of cells that do enter our G0, they're going to stay in that phase for a really long time, potentially indefinitely. And a good example of that is something like a neuron. So those neurons are obviously not undergoing apoptosis, they're not differentiating, they are just there, they are existing, they're not growing, they just exist. In terms of the checkpoints for our G0, then we've got a resting phase triggered during early G1 at a restriction point. And that's all brought about by a particular chemical, which is just a checkpoint chemical. So basically this chemical is there and then it triggers this resting phase. Do remember though that it's not every cell that will enter this, because some cells don't have G0 full stop. Things like your epithelial cells that line the gut, for example, do not have this phase. The next phase is our G1. Now, G1 is the growth phase here. So what we're going to see happening is our cells are going to grow and increase in size. We're going to get the transcription of our genes to make that mRNA, the messenger RNA. Organelles are going to be duplicated. Proteins are going to be synthesized in this biosynthesis process. And we've got this P53, a tumor suppressor gene, that helps to control all of these things going on in G1. Now, G1 itself, when we think back to our little diagram, actually occupies more than 50% of the whole cell cycle. And the reason that it takes up so much of the cell cycle is that if you're thinking about what's happening here, duplication of organelles, etc., that's not a quick process. So it's going to take time for those cells to duplicate all of those organelles that are necessary for another cell to come from it. The checkpoint we see in G1 is all to do with checking that the cell is ready to enter the S phase, that synthesis phase, so that we can then synthesize the new DNA. Our next phase of the cell cycle is the S or synthesis phase. So what we find is once the cell enters S phase, it's going to complete the cell cycle. So we're kind of at the point of no return, if you like, on our cells here. This is where the DNA is going to be replicated, and obviously we've got another video looking at that process in detail. And once we've actually got this DNA being replicated, the chromosome is going to be made of a pair of identical sister chromatids. So do remember that what we've got, a little diagram on the right there showing us a yellow and a blue chromatid, they are identical to each other, and that gives us our chromosome. Now, the S phase itself is actually quite rapid, and the reason that we have this being quite a speedy part of the cell cycle is all down to trying to reduce the risks of things going wrong. Because what we find is the quicker this happens, the less chance there is of a mutation. Because at this point where the DNA is being replicated, this is where we're susceptible to these various mutagenic agents. So think about various chemicals, radiation, etc. This is the point that it has its effect. As far as the checkpoint in our S phase goes, then what we actually find is this replication isn't just ad hoc, it's actually happening in a very specific sequence. And what we find is that the first genes to be replicated in all cells are ones we term housekeeping genes. Now, we mentioned these again in Module 6, but housekeeping genes are basically the ones that control all those fundamental processes that all cells have to basically exist. So clearly they're active in all types of cells, and because they are just the building block, if you like, of every process that can take place in a cell, they're the most important, so we replicate them first. Any of those genes that are normally inactive in a range of types of cells, we leave those to the end of our little replication cycle, so they would be replicated last in the sequence. We then enter the G2 phase, and this is quite straightforward. The cells grow. We've gone through all of the process of replicating everything. We've got our cell, but it's a bit small at this point. So you know what's going to happen? It's going to get bigger. So cells grow is what we're seeing happening in our G2. And as far as the checkpoint in G2 goes, this is all about having chemicals ensuring our cell is ready for mitosis. So we're going to see the stimulation of proteins that help make the chromosomes condense. We're going to see the different proteins that help the formation of the spindle. So all of these little aspects are going to be going on in the background while the cell is growing. Now, there is one other kind of question that comes up every so often to do with the cell cycle, and it's one of these maths-based questions, because hopefully we remember that on your paper you are going to have a proportion of questions which are going to be purely maths-based. So I've given you a little example here. Student observed the prepared slide of an onion root tip under a microscope. Total number of cells in the field of view was 265. Then we've got a little table showing the number of cells in interphase, prophase, metaphase, anaphase and telophase. And then the cell cycle takes 20 hours. The number of cells visible is proportional to how long each stage of the cell cycle lasts. Calculate the time taken for prophase to occur. Give your answer in minutes to the nearest whole number. So, first thing we need to do is identify the correct phase. So this time we're looking at prophase. So here we go. We're on this row of the table first of all. Now we've got 42 cells in prophase. We then need to work out how many cells we have in total, and it tells us in the question 265. So we are going to do 42 divided by 265 first of all, and then we obviously need to work out the length. So we've got our proportion here. We then need to multiply it by the length of time, and this is going to be obviously 20 hours in total for our cell cycle, but it asked us to give our answer in minutes. So what we've got to do, 20 times 60 times by our 42 over 265, and that's going to give us, if I just do a quick calculation, 42 divided by 265 times by 20 times 60 gives us 150. 90.18, blah, 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 blah. Now, it does also tell us in the question to give it to the nearest whole number. So all we've got to do is have a little look, because it's 190.1, then to the nearest whole number, it will be 190 minutes. So don't forget, when you are doing calculations on your papers, please show the working, because that way, if you've had a fat finger moment on a calculator use in the exam, or just a moment of stupidity under the stress of an A-level, then you will still get the marks even if your final answer is wrong. You're going to get some of those marks for the working if you've written it down. If all you've done is type it into the calculator, write down whatever it told you on the screen and hope for the best, if you mistype something, if you write something incorrectly, you get nothing. So make sure you set your working out nice and clearly so we can see exactly what it is you're doing, and therefore hopefully give you most of the marks, if not all of the marks. As always, I hope you found this video useful, and of course don't forget to subscribe to the channel so you can see when the next video is uploaded, and head on over to the website where you can find a range of other resources to help you in your A-level biology studies.